Just by a little bit of by way of background, I now have the privilege to work with this phenomenal group of scientists and developers to develop BD Catalyst as a way to advance population research in the future. I really believe this is the future of population research and the way that it needs to go. And we're so pleased to be able to develop platforms to really accelerate and facilitate scientists coming on board and doing their work. This is very close to my heart for a number of reasons. My research career was as a professor of genetics and biostatistics at Washington University Medical School. And my research trajectory was in cardiovascular disease for all those years, looking primarily at obesity and metabolic consequences and how they affected cardiovascular disease risk. So I am very, very well acquainted with population research and working with large data sets and kind of the needs of that kind of science and what you need in order to move that forward. While at Washington University, I was also a mentor with the PRIDE program that is led by Drs. Victor Davala and DC Rao. And so I've had experience for many years in working with mentees. And that's one of the things that gives me the enthusiasm to want to speak to you today about using BD Catalyst for your population research. In interacting with various mentees, I found that there were some needs that they had. They often came with interesting new ideas, hypotheses that they wanted to test and so forth, but there were some challenges in terms of where do I get the data set? Rarely did they have their own data sets because, of course, epidemiological studies are large and they're difficult to collect. And how to access existing data sets, how to access computing, and how to construct that study were all challenges that they faced as they tried to develop their research protocols and their research questions. And so we believe that BD Catalyst offers some solutions to those kinds of barriers, and that's what I'm going to talk to you about today. So this cartoon is just kind of a high-level snapshot of what is BD Catalyst. So BD Catalyst is depicted here in the center of this slide as a cloud because it is in fact a constructed cloud computing environment. In that environment, we host many different kinds of data sets, such as those depicted here, whether they be clinical, EHR, epidemiologic, genomics, and now other omics as well. And imaging data sets are becoming very hot topics in terms of looking at them and trying to identify new features. So we have ingested many of those data sets. We host the individuals, the researchers that come in. And in this constructed environment, we have security boundaries to make sure that everything is safe and sound and there aren't any risks to the data. But we also offer a bunch of tools and workflows and statistical packages and strategies and search tools to find data sets that are relevant and even AI tools to help mine these data sets. So this kind of an environment is also very hospitable to collaboration, as you can share your workspaces with your collaborators. You can be looking at the same data sets and talking and so forth. And so the idea was that if you got all of these components together in one place, in one cloud environment, it would accelerate the pace of discovery, leading to the new biology, which is the core of all of human science going forward. So that was the thinking behind it. So what does BD Catalyst bring to the table? Well, first of all, which was big for me, there's no computer system to manage. We managed our own computing system when I was doing this kind of work, and it was expensive and it was a distraction from the actual science that I wanted to do. And so this is a managed environment. You need nothing more than a desktop or a laptop computer and a solid internet connection, and you can log right into that environment and have everything at your fingertips. The ease of that is unsurpassed. We've also ingested many of these large valuable data sets into our environment already, and there's no need to download them and manage them. when one downloads data sets, there are often challenges. You have to make sure that it downloaded correctly, that there were no truncations or errors and so forth. And all of that is eliminated by this approach, which really enhances reproducible science. We all know that everyone is referring to the same data sets, and we're not having various versions out there across different compute systems. There is access to high-value data sets. As I've already mentioned, we have ingested many of the high-value ones like Framingham, Mesa, Cardia, and so forth. I will review the list a little bit further on, but it's already there. So once you get authorization to access those data set, it's the flip of a switch for that data set to be available in your workspace to continue your project. Computing on the cloud also has the distinct advantage that it's scalable. So if you only need a little bit, you're running some predictive regression models or Kaplan-Meiers or something like that, those go very quickly. The costs are very low. If you're trying a new genome, a genotype calling algorithm across 200,000 subjects, it'll be a little bit more. But the point is that you don't have to think a priori, how much compute do I need, and then have that hardware and software on board. This is a completely scalable environment, and you're only charged for what you use. In order to get you started, NHLBI is now running a pilot credits program, which allows you to come in and have a fair amount of computing to look around, explore data sets, and for many kinds of projects that I can think of, it should be enough to even complete a project. So there is a way to get going, but the expectation would be that going forward, that one would write in one's grant budget the funds to cover compute credits going forward. While in the environment, you have full access to many of the tools and workflows that have been developed, and budget calculators in case you're looking for how should I budget this in my grant and so forth. And so all of those tools are readily available to help support your programs. We have 24/7 self-service support, but also we have live office hours. And we also have a variety of YouTube videos. So at the bottom here, you see a pointer to a YouTube video. And so there's a lot of ways that one can explore the ecosystem and get answers. But we also have live people that are there to make sure that you can move forward with your project and you're not blocked by something. And by coming on board, you also become a member of the research community. Many of the fellows and the users that we've had on board have created relationships, and they work with each other. They offer each other support. They offer each other solutions. They discuss problems and so forth. And so we offer that kind of community, which can be very beneficial. As Stephanie pointed out, that sort of networking can lead to very interesting collaborations and opportunities to do more research. So as an early career scientist, I think that one of the challenges is how do you get started? And I think for the PRIDE mentees, this is exactly what the program was designed for, is to help you consolidate your thinking and to kind of think about how to build a research program and how to ask the right questions and how to move forward. And we believe also that BDC offers some of the solutions that if your project happens to be in the area of population science, we can really help in accelerating your progress there. So that there are other issues and barriers that you don't need to worry about. And I've already covered some of them, and I will be touching upon more of them as we go through these slides. This sort of acceleration in the early part of your career can be very helpful, as there's often a race to your first R01 or to get your first funding opportunity. And so not having to deal with some of these other kind of distracting blockers can really help accelerate your progress. It can help accelerate your progress for public health research, primarily because we do have so many ingested data sets that can be used as pilot data sets to evaluate your hypotheses, to generate preliminary data, and so forth. And so this allows you to have an interactive way to strategize how you're going to conduct your research and how you might want to write your grant proposals. So this can be very, very helpful in terms of moving your research program forward. So let's start with you. And like I say, this has been my experience with many of the mentees that I have worked with. They come in, they have an area of interest, maybe even a hypothesis that they wish to test, just like Stephanie came in with her hypothesis about how discrimination could actually affect inflammatory processes that are important to the development of cardiovascular disease. And so you want to explore that hypothesis. So you may wish to know, well, are there data that have collected the variables that I need? How big are those sample sizes? Are there enough subjects so that I have an adequately powered analysis and I can be successful? And all of those things can be readily assessed on BD Catalyst with the data at hand. One of the other features that I think is important to keep in mind, especially if you're early on in your career, there can be some movement, okay? So maybe you're moving from a postdoc or a fellowship to a faculty position, or you're changing institutions, or even if you're going on vacation. The beauty of working on BD Catalyst is that your environment doesn't need to shift, not your compute environment. As long as you have that laptop and that Wi-Fi connection, your entire workspace, your data, your work, your results move with you. You don't have to like transfer files and that kind of thing. Even if you go visit a colleague at another institution or something like that, you can just sit down and access your data and move on with your project. Even if you're a Pride mentee and you're for the summer at your institution, you have your whole workspace and all of your data assets with you right there. And we think that that's a real advantage of not being tethered to a local compute system or to a disk where your data sit and so forth. It's so mobile and it's with you wherever you want to go. It's also very useful to have state-of-the-art data science tools. You always want to be using the latest and greatest, the most innovative and the most efficient tools possible. We here have the advantage that we don't have to worry about versioning and so forth, because we have a lot of those tools already ingested into our toolboxes, and you can access those. There have been tools that have also been contributed by former users or by fellows that have come through. And so there's no need to reinvent the wheel in some cases. Just for example, we had a couple of people coming through who were interested in gene by environment interactions. And I mean, you could write those programs by yourself, but they contributed all those workflows and code to the repository there. And so you don't need to reinvent the wheel. You can actually accelerate your progress to analysis by simply using those coded programs and substituting in your data sets, your variables, and so forth, and off you go. So this keeps you very current and very agile in terms of really quickening the pace of the research. Again, I think one of our best assets is access to existing high-value data sets. BDC hosts many of the high-value legacy data sets and also ongoing data sets, data sets for which variables and phenotypes and assessments are still being conducted. Eventually, the goal is that BDC will be the repository for all data sets. So it'll be one-stop shopping for all those data sets, and with the idea that they can be interoperable, which I think really opens some really wonderful opportunities for data mining across different data sets. As a matter of fact, just to give you the further vision, we're looking for that interoperability across many different NIH platforms. So there can be broad interoperability and a broad look at different biological pathways that may be relevant to different diseases. Access to these data sets is controlled, and this is out of respect for the patient and subject consents that were signed. And so there are applications to dbGaP that need to be made. They are brokered by us. There's a data access committee that will just look at your credentials and look at the proposed project and grant access. Once you have that access, as I said, it's fairly simple to flip the switch where all of a sudden then you'll find that the data sets you applied for, you can now access. They can be located into your workspace and you're off to the races. But even before you go through that step of approval, you might be thinking to yourself, well, which data sets should I ask for access to? I'm not sure which ones have the variables I want or whether there's enough sample size for it to be worth it. Well, there's a way that you can start looking the data and assess all those kinds of questions so that you can fine-tune your application and go for only those data sets that are really relevant to your research project. So here's kind of just a list to tantalize your imagination of the data sets that have already been ingested into BD Catalyst. There's information on Recover, which is an ongoing study on long COVID. And this is still ongoing. So because we're still wanting to know what are the out effects of people that have long-lasting symptoms from their infections. But all of these data are starting to become available and are available for mining by other investigators. We have COVID-19 data, which are clinical trials that are available for analysis and reanalysis. We also have many of the COPD gene data sets. Those are under the TopMed banner, but then we also have Lung Map, which is a molecular atlas of lung development. So there's a lot of imaging data there. We have also brought together many of the sickle cell disease data sets. This is one of those areas of research where having many different data sets can really be helpful in terms of sample size and the ability to verify hypotheses across different studies. We have all the TopMed data. And I'm kind of surprised and blanched at the idea of calling TopMed a study because it actually includes over 85 different data sets with a broad distribution across different ethnic groups. There's Hispanic data, African American data, Asian data, European descent data, across many different kinds of heart, lung, and blood phenotypes. So it's extraordinarily rich. It's just that all of these studies have come under the umbrella of TopMed and have received whole genome sequencing and now many different other kinds of omics, making this repository incredibly rich. Not only do we have all that ancillary data sets, but many of those data sets are longitudinal. And that opens up really phenomenal opportunities for looking at causality and to see whether risk factors that are measured at young ages are predictive of disease later in life. So that has profound implications for prevention strategies and identification of populations at risk. So there's a lot of opportunity among those data sets. So there are many ways that one can explore those data sets using the tools that are available on BD Catalyst. I've already mentioned in my talk there about TopMed data sets, so I won't belabor that too much more. Recover, as I said, is long COVID. So those Recover data sets are becoming more increasingly available as the study moves forward. And we also wanted to mention that if you have your own data set and you just want to come into the environment and use the same tools that a previously published paper used in order to eliminate that as a source of variability so you can compare results directly, one could do that. One may also wish to bring in your own data to combine with other data sets that may be available on BD Catalyst. So if you have your own data, we can readily help bring those data on board. The first assurance is that there will be security. They will exist in a secure workspace with controlled access that will only be available to other collaborators that are specified by you, the principal investigator, that meet the requirements of your data use agreement. So just by bringing your own data does not mean everybody in the ecosystem can see them, only the people that you specify. It's an easy way to collaborate across institutions and locations. And you can then, as I mentioned, use your data with the BDC analysis tools and workflows that are existing to provide comparability, perhaps with previous research or other ongoing research. So how do you access the data on BDC? First, there's open access that you don't require a login. You can just go in there and you can just browse the data sets. And the two main features of data sets that you can look at are the variables that are collected. So you can say, hey, do these data sets have the social determinants of health? Do they have the discrimination index, as Stephanie used in her research? And so you can identify those data sets that have the information that you're looking for. The other thing you can assess is the number of subjects that are typed for that particular information and other information like outcomes and so forth that may be of interest. And in that way, you can do power calculations and an assessment of whether there are sufficient data in order to meet the requirements for your study. And this requires no access, no special access. You can just come on board and sort of build an idea of what you want to do. Once you know that, you would go ahead and apply for access to the data sets that are relevant to your question. And the authorized access, it's not via ERA Commons, it's actually from the Data Access Committee at NHLBI, but you log into the system using an ERA Commons login ID. So the authorized access now allows you to touch individual data. So you can actually look at the distributions of data, look at the distributions by sex or by race or whatever, and get deeper knowledge of the distributions of your variables and to conduct analyses. So one of our messages is don't be constrained. Don't be constrained primarily by those nuisance barriers that can help slow you down. One of our mottos in the development of BDC is to help you accelerate to aim one. And that was our shorthand way of saying all those things that you need to do in order to start doing your analysis and get going. We're trying to smooth that out for you so that you don't have to deal with those complications. So we have free pilot cloud credits, which can help you get started immediately, okay? There's no need to download anything. All the data sets are there. The tools and the workflows are there. There's no need to manage licensing for any of those tools. Those are all centrally managed in BD Catalyst. You use only as much compute power as you need for your program. The budget is sized for your programs, and you can access it from anywhere. You can take your laptop on vacation to Alaska and still have access to all your data and compute, whatever. So in this way, I think it gives a great deal of freedom, and it breaks down a lot of the barriers to getting started. It's easy to collaborate too. Our workspaces are on Google, Amazon, or Azure. That's not particularly important for collaboration, but in those collaborative secure workspaces, as I've mentioned, you can specify who are the people that you would like to have there to view results with you, to comment, to access files, maybe to do ancillary analyses. So it's a fantastic environment in which to collaborate. You can also get a lot of support for your research journey by using the ecosystem, like I said, with the help desk and the community that we have on board. So there are people around that can help you get past any sort of blockers you may be experiencing. Yes, you do need state-of-the-art tools workspaces. I think we've covered that. Oh, here is just sort of a sampling of the kinds of tools and workspaces that we are thinking about. So I think for statistical analysis, it's useful to realize that we have a SAS on board. A lot of people are familiar with the statistical analysis system and RStudio. So you can do R programming for statistical analysis. You can also program in Jupyter or Python, whatever kinds of other tasks that you wish to accomplish. One of our primary charges was to enable TopMed for genomic analysis. So we have a full suite of tools for doing genome-wide association studies that have been offered by two of the main groups in the United States conducting those kinds of analyses. Also, the variant calling methods for genomic data have been contributed. They are the state of the art. So if you've sequenced a small sample or a sample of your own and you want to use those data sets with the kinds of information that's leveraged from TopMed, it's possible to do that. We have developed a suite of AI-assisted machine learning models for image visualization and analysis. So those can be CT scans, it can be X-rays, what have you. Those are very rich data sets that have yet to be analyzed deeply. We have user-contributed workflows and so forth that you can leverage. And a lot of these workflows have been put into Dockerized apps. And so you can go to DocStore and download many other kinds of tools and approaches that you might be interested in looking at. We also have a fairly comprehensive search user interface, which allows you to, it also has a semantic metadata. So you can put in words like, I'm looking for African-American men with hypertension, and it will help you find the data sets that have those kind of data in them. So there are many different ways to try to identify the data sets with the variables of interest and to help you move that forward. As I said, if you need support getting in this environment, we have support for you. We have a ton of written materials and guides. I would advise people who are interested to have a look at our website, which we will have a link to somewhere. And there's a lot of information there on data sets, workflows, how to, and so forth. We have also a big collection of YouTube videos on how to do things and how to deal with certain kinds of problems or how to program in SAS or what have you. So there's a lot of tutorials there. We've done many of these community hours and webinars, and so those are recorded and they're also archived so that they're there for your listening pleasure. And we have virtual office hours and drop-in sessions. So you can always get in touch with a person if you really need a personalized approach to help you get past whatever it is that's vexing you. Help desk assign online forums so you can put your questions in chats and so forth, and other people can maybe weigh in and help out things. And you can make an appointment with a subject matter expert if you really need to know something about a particular data set or a phenotype that is important to understand. So as I've mentioned, there are pilot credits that are available. Once you join the community, you can apply for the pilot credits, and they're quite generous actually. And we have policies about how to do that. No more than I want to explore the ecosystem is needed in order to secure this resource. So we want to make sure that there's no barrier for you all to come on and explore and see what can be done in this world of BDC. In exchange for pilot credits and for use of BDC, what we ask for is acknowledgement in any publications or presentations that are made. This is important for tracking our progress and making sure that we understand how far our reach is and how impactful we're being in terms of helping out researchers. And guidelines on how to cite BDC are on our website. It's very easy to do. So we would welcome you here with us. Our community is very diverse and broad, and it includes early stage as well as experienced researchers. Some of them are from these consortium projects. Some of them are individuals that are here to do secondary analysis of existing data sets. We have principal investigators, we have analysts, we have all sorts of people. On the BDC side, we have computer scientists, data scientists, and developers, and so forth. So we have a lot of people that can help also if there are problems or tasks or tools that need to be created in order to move things forward. So if you would like, there is no obligation. There's no hook there, but if you just want to join our community, please click on that link. We'll just take your name and email address, and you are ready to get started to explore our resources and claim your pilot credits if you'd like to forward and see what it can do for your research program.